Hello everybody, I am Deepika from LearnoHub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. Students, today we are going to start with the next chapter of class 10 biology ICSE, that is photosynthesis. So we will look into the details of this chapter. So we all know what is photosynthesis, but it's time to get into the depth of photosynthesis. So let's get started. So starting with photosynthesis. So photosynthesis is a very significant life process in plants. If there is no photosynthesis, so there will be no food, right? So what are plants called? Plants are autotrophs, right? So they are called as autotrophs, the organisms which prepare their own food. So they are called as autotrophs. So plants are autotrophs. They perform photosynthesis and in turn they give us food in the form of starch, in form of carbohydrates, in form of sugars. And during this process, they also release a life-supporting gas, which is oxygen. So they give us oxygen, right? So they provide food to all the living beings, whether they are herbivores, they are carnivores, they are omnivores. So we know that if we talk about the different types of animals on the basis of what they eat, so they are herbivores, the ones who only eat plants, right? And there are carnivores and omnivores. So omnivores, they eat plants as well as animals, and carnivores, they are just dependent on animals for their food. But think of a scenario when there are no producers. So carnivores, if we take lion, right? So lion feeds on a deer and deer is a herbivore. So if lion wants to feed on the deer, so there has to be some population of deer so that lion can get its food, right? So basically, directly or indirectly, all the animals or all the living organisms, they are dependent on plants, right? So if we have plants, right, then there is a deer and there is lion, right? If there are no plants, deer will not get food. If there is no deer, lion will not get food. So basically, deer has to eat plants so that this deer population can survive, and then only lion will be able to eat that herbivore. Correct? So I hope this is simple. Now, it provides a life-supporting free oxygen gas in the atmosphere for breathing. So it becomes that oxygen gas is very, very essential, right? So if there is no oxygen, we won't be able to breathe. So many cellular processes won't happen, and ultimately we are going to die. Correct? So I hope the basics are clear. So let us move on to our next topic, coming into more depth of what is photosynthesis. Now the next topic is what is photosynthesis. So photosynthesis is basically a process by which living plant cells containing chlorophyll. So photosynthesis happens only in those plants which have chlorophyll. They produce food substances like glucose, like starch, from the raw materials which are carbon dioxide and water by using light energy, right? So what happens, there are four important raw materials for a plant to photosynthesize. So the first one is carbon dioxide, next one is water. So plants get carbon dioxide through their stomata from atmosphere, right, from air. Now there is the second requirement, that is water. So roots of a plant, they absorb water from the soil, and then in the presence of the pigment called chlorophyll, they trap sunlight and they convert these carbon dioxide and water into sugar, or you can say starch or glucose, and they release oxygen gas. So this oxygen gas is a byproduct which is very, very helpful in the survival of all the living beings, right? So what is the importance of photosynthesis? So the first importance is food for all. So all the living organisms, directly or indirectly, they depend on plants and they get their food only when plants are there. Second is oxygen to breathe in. So when plants photosynthesize, so we studied that they release oxygen gas. So this oxygen gas which is released into the atmosphere, it is taken in by the living beings and the cellular processes happen inside the living organism, right? So both it gives us food as well as it gives us oxygen. So photosynthesis is very, very important to all of us. So the next is chlorophyll. So we are talking that plants, they contain chlorophyll, right? So this is the vital plant pigment which is responsible for photosynthesis. So what is this chlorophyll? If we talk about chlorophyll, it is made up of two words. So the first one is chloro and the second one is fill. So chloro means green and fill means leaf. So this is the green pigment that is contained inside the cell organelles which are very microscopic and they are called as chloroplasts. So where is this chlorophyll present? It is present in the leaf. And what do these leaves have? They have chloroplasts, right? So these are the organelles which are chloroplasts, correct? So I hope this is clear. Now we are going into more detail of this cell organelle which is only and only present in plants, not in animals, and that is chloroplast. Now the next part is that organelle, that cell organelle which contains chlorophyll, and that is the chloroplast. So this is the structure of a chloroplast, where you can see this is a double membrane structure, right? So it has two membranes. So one is this and another is the outer one, right? So these are the two membranes, the outer membrane and the inner membrane. So the chloroplast envelope has two membranes, inner and outer, right? So inside these two membranes there is this fluid part which is called as stroma, as we call it in a cell, a cytoplasm. So that fluid part inside this these two membranes or inside a chloroplast is called as stroma. Now this stroma has flattened sacs. So do you see this flattened sac? This is one. So one is lying below this, one is here, one is here. So these flattened sacs, they are called as thylakoids, correct? Now these thylakoids, this is one thylakoid, this is second, this is third, this is fourth, and so on, right? So when these thylakoids, they pile over each other, so that is called as granum. So this is granum, right? Now there are some interconnecting bars. So you can see over here, this is one grana, these are the thylakoid stacks, and the complete one is called as grana, and this grana is connected to this granum and with a interconnecting bar kind of a thing, a kind of a rod, right? So that is called as a fret, correct? So this is the structure of a chloroplast. So what are these chloroplasts? So these are the minute oval bodies which are present inside the leaves of plants, and they are double membraned bound structures, right? So they have two membranes, the outer one and the inner one, correct? Now if we talk about a per cell, so there are 40 to 50 chloroplasts per cell. We are not talking about per leaf, we are talking about per cell. A single cell inside a leaf contains 40 to 50 chloroplasts. Easy, clear? Now this pigment chlorophyll, where is it present in this chloroplast? So we are saying that chlorophyll is present in the chloroplast. So where is this chlorophyll? Where is this pigment inside the chloroplast? So these chloroplasts, they have thylakoids, and the walls of these thylakoids, they contain chlorophyll, right? So these walls of these thylakoids, they contain what? They contain the pigment called as chlorophyll. So now this chlorophyll is made up of what? So it is composed of carbon, hydrogen, oxygen, nitrogen and magnesium. So these 5 things are involved in making one chlorophyll, right? Clear? Till now it is clear. So we studied the structure, we studied where is it present, and now what is this chlorophyll pigment made of? So it is made up of carbon, hydrogen, oxygen, nitrogen and magnesium. Clear? Easy. Now these chloroplasts, where are they present on the leaf? If we talk about a leaf, right, so where are these chloroplasts present? So these chloroplasts are present in the mesophyll cells. So where are they present? They are present in the mesophyll cells. So we have already studied all of these, right? So mesophyll cells are present in the leaf, and they are located between upper and lower epidermis of the leaves, right? So these chloroplasts are present in the mesophyll cells, which are located on between the upper and the lower epidermis of the leaf, correct? Now the next thing that we have already studied is that when we were studying the opening and closing of stomata and many other things, so that time we found that stomata, they are guarded by some bean shaped cells, and we call them as guard cells, right? Now these guard cells, they contain what? They contain these cell organelles, organelles which are called as chloroplast, and these chloroplasts have what? They have chlorophyll, right? So now when I was telling you that a single cell, it contains 40 to 50 chloroplasts, right? Now we are talking about a leaf. So if we talk about a leaf, if we talk about per centimeter square, right? So per square, not centimeter, per millimeter square of a leaf, it contains around 5 lakh of chloroplasts. So how many organelles are there? How many chloroplast cell organelles are there inside a leaf? So if we talk about per millimeter square, so there are about 5 lakh chloroplasts, correct? Now chloroplasts have chlorophyll. Now this chlorophyll is not just a single type of a chlorophyll, but basically there are 9 types, and out of those 9 types, chlorophyll A and chlorophyll B are the most abundant chlorophyll, and they are very helpful in trapping the solar energy. Clear? So I hope this part is clear, where we studied about first the structure of the chloroplast, and then we studied about chlorophyll, where is it present, what is it made up of, and what are the different types of chlorophylls, right? So where are these chloroplast cell organelles present? So they are present between the upper and the lower epidermis of the leaf, correct? Now the next question that comes into our mind is, why is chlorophyll green in color? So we heard that these chloroplasts, they have chlorophyll pigment, right? Now this chlorophyll pigment, it captures sunlight, it traps sunlight, correct? Now sunlight has a very broad spectrum, it has different colors, right? So if we talk about what color is being absorbed by chlorophyll, so it basically absorbs red and blue light, right? So it is absorbing colors from both the ends of the visible spectrum. Now in the visible spectrum, there is red light also, there is blue light also. So it is basically absorbing this red and blue light and reflecting the green light. So that is why, since it is reflecting the green light, so we see these chlorophyll as green in color, right? And this pigment only gives green color to the leaves. So chlorophyll absorbs light at both the ends of the visible spectrum. It reflects away only green light. That is why chlorophyll appears to be green and hence gives green color to the leaves. And red and blue light are the most effective lights for photosynthesis. So for photosynthesis, we do not need green light. We just need red and blue light from the visible spectrum. So now talking about the chlorophyll, do you think that too much of light actually destroys chlorophyll? So that is true. When there is too much of exposure of light to a plant, so that destroys the chlorophyll because it disintegrates the chlorophyll that is present inside the leaf. And also if the plant will not get enough sunlight, so in that case also there will be no chlorophyll production. So basically the production of chlorophyll, it depends on the exposure of light. So just try to observe one thing. So if there is some grass which is growing under a shade, right? So we can see grass everywhere around us, but just try to observe the grass which is growing under the shade. So there you can actually see that that grass is not green, it is actually yellowish in color. Why? Because there is that grass has no exposure to sunlight, and because it has no exposure to sunlight, so there is no synthesis of the pigment called chlorophyll. Clear? Now the next part, that is regulation of stomatal opening for letting in carbon dioxide. So now we are talking about photosynthesis. So for photosynthesis, one of the important raw material is carbon dioxide. So this means plants have to take in carbon dioxide, right? So how do they do that? So they take in the carbon dioxide from air through the small tiny minute pores or the openings which are present in the stomata, which are present in the leaves, and these are called as stomata. So the upper and the lower epidermis of the leaf, both of them contain stomata, and it is said that the lower surface of the leaves contain more number of stomata as the upper surface of the leaves, right? So basically what happens when the main function of stomata is letting in carbon dioxide while they have to perform photosynthesis. So what happens now when during the day, what happens when there is sunlight? So plants, these leaves, they open their stomata which allows carbon dioxide to enter into the leaf, right? But during the night time, during the dark, when there is no sunlight around, so at that time these openings, these minute pores, they close. Why? Because there is no photosynthesis, so they do not need carbon dioxide anymore, and there is no generation of oxygen gas, which is the byproduct of photosynthesis, so they do not need that gaseous exchange. But what happens along with this opening of stomata? So when this stomata opens for the gaseous exchange, at that time transpiration also happens. So it is not wrong to say that the transpiration, that is the loss of water in the form of water vapors from the leaves, is the price that a leaf or a plant has to pay to perform photosynthesis. Correct? So as you can see this, we have already studied it in detail in the chapter transpiration. So these are what? This is the open stomata, this is the closed one, right? So these are the guard cells, right? So these kidney shaped cells are the guard cells. This is the stomatal pore. These outside cells are the epidermal cells. This is the inner wall of guard cells. This is the outer wall. So you can see this inner wall is thicker as compared to the outer wall. And now this fluid inside these guard cells is called as, this is the cytoplasm, and their cytoplasm contains chlorophyll, as we discussed, right? It also contains nucleus. So these are the guard cells. So what happens? The water from the surrounding cells, it gets into the guard cells, they swell up and they make this spindle shaped structure. Right? Now when there is no sunlight, so during that time the water moves out of these guard cells, so they lose their turgidity, and finally the stomatal pore, it closes. So this is what something that we have already discussed in detail in the chapter transpiration. So in the previous chapter, we studied that opening and closing of stomata is just because of the turgidity of the guard cells, whether the guard cells become turgid or they lose their turgidity. So that is the main reason why stomata, they open or they close. But this is not the only case, right? So there are basically the two theories which decides the opening and closing of stomata. So one is the new theory and one is the older version of the theory. So the new one is potassium ion concentration theory. So we'll discuss them in detail, and the older one is sugar concentration theory. So let's discuss each of them in detail. So the first one is potassium ion concentration theory, and let me tell you that these two theories, they are very simple. So as I was discussing in the previous chapter, while we were discussing about transpiration and then opening and closing of stomata, then that time we were talking about the movement of water into the guard cells, which makes it turgid and gives that spindle shape to the guard cells, which opens the stomatal pore, right? So according to this potassium ion concentration theory, which is said to be the most recent one, so according to this, the stomatal opening and closing depends on the generation of K positive ion gradient, right? So basically what happens when there is sunlight, so the chloroplasts which are present inside the guard cells, they start photosynthesizing. So when they photosynthesize, so they generate ATP, that is energy. So this ATP which is generated during this process, so that is used to actively pump potassium ion. So these are the adjacent cells, this is the guard cell, these are the adjacent cells, right? So that ATP actually actively pumps, putting that energy, so it pumps in what? The potassium ions. So there are potassium ions over here, so they will actively pump these potassium ions into the guard cells. Now when these potassium ions will come into the guard cells, so what will happen? These guard cells, they become hypertonic, right? So the ions are moving in, so this means the water is becoming less, right? So now what will happen when these guard cells, they become hypertonic? In that case, the more water is drawn in from the adjacent cells into the guard cells. When more water is drawn in that case, these guard cells will gain turgidity, so they will become turgid. When they will become turgid, they will swell up and they will take this bean shaped structure, and this leads to the opening of what? This leads to the opening of stomata, right? So it is something related to that turgidity, that hypertonicity only. Only the case over here is we have to start this theory mentioning that there are chloroplasts which are present in the guard cells. They start photosynthesizing, hence they produce energy in the form of ATP. Now this ATP brings in more potassium ions from the surrounding cells, and when there are more number of potassium ions inside the guard cells, so these guard cells, they become hypertonic. When they become hypertonic, then they will tend to gain more and more water from the adjacent cells. So more water comes into the guard cells, so they become turgid, and when they become turgid, they swell, and hence they get that spindle shaped or the bean shaped, and this leads to the opening of the stomata. Now the reverse happens during the night. Now during night time, these chloroplasts, they will not photosynthesize, they will not perform photosynthesis. Why? Because there is no sunlight, correct? When there is no sunlight, so in that case, there is no production of ATP. When there is no production of ATP, there will not be any movement of K positive ions or the potassium ions into the guard cells. Hence, they will lose water, the extra water that is inside these guard cells. Correct? So, along with that, why that they are losing water because now the K positive ions which were initially present inside the guard cells, they will also start moving out. They will leak out into the surrounding cells, and hence water will also move out. Now what will happen? These surrounding cells, they will become what? They will become hypertonic. So they will gain, they will try to get more and more water from the guard cells, right? So this is just the case of hypertonicity and turgidity that we already studied, right? So it is the movement of the K positive ions that leads to the opening and closing of the stomata. Now the second theory is the sugar concentration theory. So according to sugar concentration theory, it is the older version of the theory in which it is said that during the daytime, the chloroplasts which are present in the guard cells, they start photosynthesizing. So when they photosynthesize, they will produce what? They will produce sugar. Now the sugar concentration inside the guard cells increases, which leads to the increase in the osmotic pressure. As the osmotic pressure increases, more and more water is drawn from the surrounding cells. So these surrounding cells will give more and more water to the guard cells, and hence they swell up, and when they swell up, they gain that turgidity, correct? So which leads to the formation of this shape of the guard cells, leads to the opening of the stomata, correct? Now for closing of stomata, it is said that when there is the water content of the leaf, it becomes short. During that time, the water is withdrawn from the guard cells, leading these stomata, basically the inner wall, to straighten up. So this inner wall becomes straight, and hence this leads to the closing of the stomatal aperture or stomatal pore. Now the next topic is the process of photosynthesis. So if we talk about the process of photosynthesis, what is the primary center of activity? The primary center of activity is the mesophyll cells, right? So these mesophyll cells, they contain chlorophyll, correct? Now these mesophyll cells, they are made up of palisade and spongy cells, right? So palisade cells and these spongy cells together made mesophyll cells. Now these mesophyll cells, so when during the daytime when sunlight falls on the leaf, so these mesophyll cells upper layer, that is the palisade cells, they absorb this sunlight, and when this sunlight is absorbed, this increases, this leads to the production of energy, which is which actually helps in the formation of food or starch or sugar inside the leaves, correct? So this is what? This is the process of photosynthesis, correct? So these are the primary activity, and for this process we need carbon dioxide and we also need water. So how do they come in? Talking about water, water is absorbed from the soil, and from soil it there are root hairs absorb this water, and they through the xylem vessels, it moves up into the stem and finally reaches to each and every part of the plant through the xylem tissue, right? And now talking about the carbon dioxide, so it comes into the plant through stomata by diffusion. Now in diffusion, what happens? If we talk about carbon dioxide, so it moves from its higher concentration to lower concentration. When the stomatal aperture opens, so there is higher concentration of carbon dioxide outside the cell, but it is very less concentration inside the cell. So by the process of diffusion, this carbon dioxide enters into the leaf through what? Through stomata, correct? Now talking about the chemical equation of photosynthesis, so we have already discussed that there are 4 important raw materials: carbon dioxide, water, sunlight and the chlorophyll, right? So in the presence of chlorophyll, which is present in the chloroplasts of a leaf, right, so that it traps this solar energy, and carbon dioxide is obtained from the atmosphere. It diffuses into the leaves through stomatal aperture, and water which is present inside the soil is absorbed by the root hair. So these four ingredients, they together form sugar, they release some amount of water, and oxygen gas is released as a byproduct in this process of photosynthesis. Correct? So the water is absorbed as well as water is reformed in this process of photosynthesis. So this equation is quite important because there are many questions that roam around this equation. Now talking about photosynthesis. So photosynthesis happens in two phases. So one is light dependent phase and one is light independent phase. Correct? So let us talk about these two phases in detail. Now the first phase is the light dependent phase. So as the name suggests, this phase is dependent on light. So we also call it as photochemical phase or the light phase. So where does this phase takes place? So it takes place in the thylakoids. Fine. So in the thylakoid, so basically there are two reactions that are taking place in the light reactions. One is activation of chlorophyll and another is splitting of light. So chlorophyll, when it is exposed to light energy, so it absorbs that light energy by absorbing photons. What are these photons? These photons are the smallest units of light energy. So this chlorophyll will absorb these photons and hence they will get activated. Now there is lot of energy inside the thylakoid. So this energy leads to the splitting of the water, and this process is called as photolysis. So the water molecules, in the presence of photons, it leads to the splitting of this molecule, hence giving us H positive ions, electrons, and oxygen gas is released during this process, right? So basically photolysis is the splitting of light. Photo means light, lysis means splitting, correct? So splitting in the presence of light. Correct? So these are the two main steps of the light reactions. Now coming on to the end products of photosynthesis. Now we see that chlorophyll has been activated and the water molecules has been split. Now what happens to these H positive ions which are being produced? So these H positive ions and some electrons, they are taken up by NADP, and this leads to the production of NADPH in the presence of enzymes. Now the second thing is oxygen. So these oxygen, they together come as these oxygen molecules and they give us oxygen gas. Now comes the last one, there will be ADP. ADP will pick up the inorganic phosphate and this leads to the production of ATP, that is adenosine triphosphate. And this process where ADP is converted into ATP by addition of one phosphorus, inorganic phosphorus, so this is called phosphorylation. Now since this whole process is occurring in the presence of light, so we also call this process as photophosphorylation, right? So this is also called as photophosphorylation. So these are the end products of photolysis. Now the second phase is the light independent phase, which is also called as dark phase or the biosynthetic phase. Now calling it as a dark phase is somewhere not right, because it does not mean that this phase happens during night or this phase only does not happen during the daytime, right? So basically what is happening, the reactions that happens during light independent phase, they are not dependent on light. So what happens during this light independent phase? So there is conversion of glucose into starch and other chemicals. Now we know that during photosynthesis a lot of glucose is produced, right? A lot of glucose molecules are being produced. So these molecules together they form starch, which is a polysaccharide, and this process is called as polymerization, right? So what is it called as? It is called as polymerization. Now these glucose molecules, they are converted into starch and sometimes into oil, sometimes into cane sugar. So there are different products into which this glucose molecule is getting converted. And when is this happening? This is happening as soon as the glucose molecule is produced. There is not a much gap, time gap between the two processes, between the light independent reactions and the light independent reactions. Fine. Now let us look into the summary of the events that occur during light reactions and light independent reactions of photosynthesis. So there are two phases. One is the light dependent reactions, one is the light independent reactions. The first difference between the two is the light dependent reactions, they take place during the daytime and in the thylakoids of chloroplast, while the light independent reactions, they take place in the stroma of chloroplast. The second thing is, during light dependent phase, we have studied that there is activation of chlorophyll, and when this light energy is absorbed, there is lot of energy inside inside the cell. So it actually leads to the photolysis, that is the breakdown of water. When the photolysis happen, there is generation of some oxygen atoms, hydrogen atoms, electrons, right? So this is the H positive ions. So oxygen is given out as a byproduct, correct? So it is given out. Secondly, this hydrogen and some of the electrons, they combine with NADP and they form NADPH. Whereas these electrons, they combine with ADP and the inorganic phosphate, and they leads to the formation of energy in the form of ATP. So this is called as phosphorylation. So phosphorylation is also happening during light dependent reactions. On the other hand, in the light independent reactions, now carbon dioxide is getting converted to glucose, right, during the photosynthesis. Now during this phase, the glucose is getting converted to starch. So a lot of glucose molecules, they are coming together and they are forming the starch, right? Now during this time, the NADPH, which was being formed, so it loses its hydrogen ions and again gets converted to NADP. And in the same way, ATP also loses its electrons and gets converted to what? It gets converted to ADP and the inorganic phosphate. So this is the complete summary of the two types of reactions that happens during photosynthesis. Now the adaptations in plants to perform photosynthesis. So we have seen certain adaptations in certain plants so that they can perform more and more photosynthesis, so that they can produce more and more food. So the first one is leaf surface area. So we know that leaf contains chlorophyll, leaf contains chloroplast, and which is very essential for photosynthesis. So basically they increase their surface area so that they can trap more and more light and they can perform photosynthesis. But in some of the plants, it has been seen that the leaves are narrow. This is just to reduce the transpiration that is happening through the stomata of the leaves. Now the second one is the leaf arrangement. If you look at the plants, so if this is the solar energy, right, if this is the source of energy, mostly the leaves are like this, right? So they are basically at a right angle to the source of light, so that more and more light falls on the leaves. Now the second one is the cuticle and the upper epidermis. So it has been seen that this cuticle and the upper epidermis is transparent and it is waterproof. So this allows more and more light to enter into these leaves. Okay. Then numerous stomata. So now we know that this gaseous exchange, it all happens through the stomata. So if there are more stomata, more carbon dioxide will enter into the plants and they will be able to perform more photosynthesis when the light is present. So this is the fourth point, right? Now the next is the thinness of the leaves. Now if the leaves are thin, so they will there will be a reduced distance between the cells, right? So this will lead to rapid transport of the things into the cell, right? So the thinness of leaves is again a very important point. Then coming on to the chloroplast. Now what do these chloroplasts have? So you all know that they have chlorophyll pigment in the walls of the thylakoid membranes. So these chloroplasts, more the number of chloroplasts, more will be the chlorophyll, more will be the chlorophyll, more will be the absorption of light, right? Then comes the extensive vein system. So there is rapid transport to this vein system leads to the rapid transport to and from the mesophyll cells. Now mesophyll cells are the cells where all these things happen, all this process happens. So this extensive vein system leads to the rapid transportation of material to and from the mesophyll cells, correct? So these are the adaptations of plants to perform photosynthesis. Now the end products of the photosynthesis. So talking about the end products, so there were 3 end products. One was glucose, one was water, and one was oxygen. So what happens to these end products? So talking about the glucose first. So glucose is basically the simple sugar that is being formed by photosynthesis. So this simple sugar is either immediately consumed by plants, or it is stored in the form of starch, or it is utilized for synthesis of proteins or fats, or it is converted into sucrose, right? So these are the four end results of glucose in a plant. Now the second one is water. So water is also being absorbed by the plants for photosynthesis. It is also given out as a product in photosynthesis. So this water can be used again and again by the plants for performing more photosynthesis. Okay. Now talking about the last one, that is oxygen. So oxygen is sometimes used by plants for respiration, and it is called as, the process is called as photorespiration, right? And secondly, major part of it, the major portion of this oxygen gas, right, it is given out so that it is used by the living organisms to breathe and to perform their cellular functions, right? So actually this oxygen is not a waste product. It is a very, very useful product for the living organisms that live on earth. Now the next topic is utilization of synthesized food and its translocation. So food is synthesized in the leaves in the form of glucose. We all have studied that in the process of photosynthesis. So this glucose is formed very rapidly during photosynthesis, right? Now this glucose is not transported with that great speed inside the plant. So for some time this glucose is stored by leaf in the insoluble starch form, and that is the temporary storage in the leaf, right? And the second is what happens during night. So at night there is no photosynthesis. So during that time this starch is converted into soluble sugars. It was insoluble starch at this time. So this insoluble starch is converted into soluble sugar, and it is transported in solution form through the veins, correct? So what happens in the phloem tissue? So water will enter from the adjacent xylem tissues, and that will make this starch soluble, and hence it is transported through the phloem tissue from leaves to the different parts of the plants, and this process is called as translocation. Now when this food is in different parts of the plant, during that time it is sometimes used for getting energy, sometimes it is stored in form of starch, again it is converted back into starch. So it has different fates when it is transported or it is translocated to different positions. So what is translocation? It is basically the movement of the prepared food by the leaves in the soluble form from leaf to different parts of the plant. Now next comes the factors that affect photosynthesis. So there are some external factors and there are some internal factors. So external factors as the light, the carbon dioxide concentration, temperature and the water content. Talking about the internal factors, so what is present inside the plants that might affect the photosynthesis? So it is chlorophyll, it is protoplasm, and also the structure of the leaf. So let us discuss all of them in detail now. Now the next comes is the external factor. So we'll be starting with the light intensity and the carbon dioxide concentration. So these two go side by side. So if I say that the photosynthesis will increase as the light will increase, the light intensity will increase, to some point it is correct. So as you can see over here, this is the rate of photosynthesis, this is the intensity of light. So this intensity of light is increasing this way, right? So as the intensity of light increases, the the photosynthesis, the rate of photosynthesis also increases, right? But after a point it becomes stabilized. So highest rate of photosynthesis is reached at 0.02% concentration of carbon dioxide. Now at this point of time, that is the S dash, if we increase the concentration of carbon dioxide, then the graph again starts going up like this, and after a point again this rate of photosynthesis will become stabilized. So the highest rate of photosynthesis is reached at 0.05 concentration of carbon dioxide. So basically the light intensity and the carbon dioxide concentrations effect on photosynthesis go side by side. So to some extent light intensity can increase the rate of photosynthesis, but after that it becomes stabilized. But if we increase the carbon dioxide concentration at that point, it increases the rate of photosynthesis again, which again becomes stabilized after some time. So next comes the external factor, that is temperature. So temperature is a very important factor. As the temperature rises, the rate of photosynthesis tends to rise, but up to a certain point. The optimum temperature is said to be 35 degrees Celsius. But after 35 degrees Celsius, this rate of photosynthesis starts decreasing, and at 40 degrees Celsius, the photosynthesis stops. Why? Because this is a very high temperature where the enzymes, they degrade and hence no photosynthesis happens. But it is said that if there is a rise of 10 degrees Celsius of optimum to the optimum temperature, so that increases the rate of photosynthesis. Say the optimum temperature was 20, if the temperature rises to 30, right? So in that case the rate of photosynthesis will double. If the optimum temperature is say 25 degrees Celsius, and if the temperature rises to 35 degrees Celsius, in this case also the rate of photosynthesis will again double. Fine. So this temperature is a very important factor, but up to a certain point. If it rises above that, so in that case the enzymes, they denature, and hence there will be no photosynthesis. Now the next external factor is the water content. So we know that how important is water for photosynthesis. So it is one of the raw material that is required by the leaves, right? So in that case, if there is less absorption of water by the root, say due to the scarcity of water, or there is the damage to the root, or the second case might be the excessive transpiration. So in all these cases there is a reduced rate of photosynthesis. So rate of photosynthesis is going to reduce a lot. Why? And this will also affect the diffusive capacity, that is the carbon dioxide intake by the stomata. So at this point of time, when there is water scarcity, stomata will close, right? When stomata will close, carbon dioxide intake will also come down, hence there will be no photosynthesis, or the photosynthesis rate will decrease. So it is said that only 1% of the water that is being absorbed by the roots is used for photosynthesis, and rest all the water is given out in the form of transpiration. Now the next are the internal factors. Internal factors, we have three factors: chlorophyll, protoplasm, and the structure of the leaf. So talking about chlorophyll, if in any case we know that what is this chlorophyll made up of? Carbon, hydrogen, nitrogen, oxygen, and magnesium. In any case, if there is lack of any of these nutrients, there will be no formation of chlorophyll. Hence, when there will be no chlorophyll, the formation of chlorophyll will get hampered. So that will directly hamper the process of photosynthesis. So in that case, the rate of photosynthesis will go down because there is no chlorophyll to trap the sunlight. The next one is the protoplasm. So protoplasm is the living content of the cell. It has a lot of water. So in any case, there is dehydration of protoplasm. So there is loss of water from the protoplasm, or there is excessive sugar concentration inside the protoplasm. So that will also decrease the rate of photosynthesis, right? And in the last one, the structure of the leaf. So structure of the leaf, so the distribution of the stomata, distribution of chloroplast, the structure of the leaf, all these affect the rate of photosynthesis. Broader the leaf, more will be the photosynthesis. Narrower the leaf, less will be the photosynthesis. Thicker the leaf, thick cuticle will also reduce the rate of photosynthesis. Fine. So I hope this is clear. The internal factors are clear to everyone. So let us talk about some more minute things that we should know about plants and photosynthesis. So from dusk to dawn, what all are the natural changes that are happening in the environmental conditions and photosynthesis? So like we said, oxygen is very, very essential for all the living organisms, right? But in case of plants, we just talk about photosynthesis, where we talk that they take in carbon dioxide and they give out oxygen. But that is not true. For respiration, plants also take in oxygen and they give out carbon dioxide. But during the daytime, this uptake of carbon dioxide is lot and release of oxygen is very less. So the release of oxygen is more, right, than the uptake of oxygen. So in that case, what happens, it is said that plants are taking in carbon dioxide and they are giving out oxygen, but that is not true, right? So they also respire, and during the day, since the net output of oxygen is high, so we say that during the daytime plants give out oxygen, right? But during the night, when there is no photosynthesis, so around a plant, if you see, if you observe or if you measure, the carbon dioxide concentration is going to be more as compared to the oxygen concentration. So plants also respire like the other living beings that live on earth. Now talking about the photosynthesis at night. So does that happen? Like, is there something like some plants do photosynthesize at night? So that is true. Some plants have a different mechanism for photosynthesis that is called as CAM pathway, and this is what you are going to study in your higher classes. So some plants, they perform photosynthesis using this pathway, the CAM pathway. Now this CAM pathway is helps them to photosynthesize, and hence they release oxygen during night. So what are those plants? So one, you have guessed it right. So this is aloe vera. So aloe vera plant, then people plant. Then the next one is Tulsi plant. So all these plants, they photosynthesize at night time, not during the day. Okay. Now the experiments on photosynthesis. So we know that there are certain important things that are being used up by the plants for photosynthesis, and there are certain things which are being released by plants during photosynthesis. So we can actually check all of those. So the very first thing that we need to do over here is to de-starch our leaf. For all the experiments, first we need to know how to de-starch the leaves, and that is what is de-starching. That is the removal of starch from the leaves. So in that case, pick up a plant, keep it in the dark for 24 to 48 hours. So during this time, the leaves of this plant, they will get de-starched, right? So they will get de-starched. So this de-starching will actually leads to no starch inside that leaf, and hence we can easily perform our experiment. So what is this de-starched leaf? So one leaf that is free from starch. Okay, so this is what we have to do before we start our experiment. So we have to keep our plant for 1 or 2 days in a in a dark room. Now the question over here that can be asked is why to kept in dark? So that all the starch is being used up, all the starch that is in the stored organs, that is present in the leaves, it gets used up by the plant, and that plant becomes free from starch. Okay, so let us now discuss how do we test the presence of starch in leaf. So to test the presence of starch in a leaf, we perform this test which is named as iodine test. So in this case, we take up a leaf, we dip that leaf for a minute in a boiling water, and after that we remove that leaf from there, put it in a methylated spirit, or you can say alcohol, you can put it in alcohol, and put this beaker containing alcohol and that leaf in the water bath, and now allow that water to boil. So keep it for some time. Now after some time you will see that that leaf becomes pale white, right? So this means that all the chlorophyll from that leaf has been removed, so it has lost its green color. And why there is green color in the leaf? Because of the presence of chlorophyll in the leaf. Now that chlorophyll is being used up, so hence that color will go, the leaf will have that pale white color, and due to which, and now the leaf has become very hard and very brittle. So now we will put it in a hot water again, so that it gains that softness, so that we can perform our experiments. Now after putting it in the hot water, spread the leaf on a dish, put it on a dish. Now pour the iodine solution. So when you will pour the iodine solution, there will be two possibilities. The one is the color blue black coloration, which shows the presence of starch, and the second is brown coloration, which shows there is no starch present in the leaves. So this is such a simple test to perform, and go anywhere, perform or starch test on any of the sample. So what experiment you will do? You will perform this iodine test only. Right? Now the experiment 1, when we need to show that chlorophyll is essential for photosynthesis. So how do we do that? So we'll take a plant which has variegated leaves, like a croton plant. So you must have seen the croton plant which has the colorful leaves, right? A leaf which has different colors. So basically, so now what we will do, we'll take that plant, we'll keep it in dark for around 2 days, right? 24 to 48 hours, and then we'll keep it in sun for 4 to 5 hours. Now after 4 to 5 hours, so why we have kept in dark? So that first we can de-starch all the leaves, we can make that plant starch free, and we know that why. Right? Now we have kept it in sun for 4 to 5 hours. So after 4 to 5 hours, we'll pluck a leaf, we will mark the areas, we'll make an outline on a sheet of paper marking the green areas, the white areas, and then we will perform that iodine test on this particular leaf, and on pouring that iodine solution, we will find out that the portion of the leaf that was green has now turned blue black, right? And the portion of the leaf that was white has turned brown. So this means this blue black color over this green color shows the presence of starch, and the white portion did not have green color. So this means it doesn't have chlorophyll. So there is no photosynthesis that has happened in this white portion. Fine. So I hope this is clear. And how to perform the iodine test, that we have already discussed. Boiling the leaf, then boiling it in a methylated spirit, and all those points, right? So this shows that chlorophyll is essential for photosynthesis. The areas where there was no chlorophyll, there was no starch formation also. Now the experiment 2 is to show that sunlight is necessary for photosynthesis. So we know that sunlight is also necessary. So let us prove that. So we will take a plant, we will first de-starch the plant. So I am not again and again explaining how to de-starch and how to perform that iodine test, but I know now you are very clear. We'll keep it in dark for some time, for 24 to 48 hours. So that will lead to the de-starching of that plant leaves. Now we'll pick that plant, correct? We'll place it in sun, but before that we'll take a black sheet and we will make some design over it, say the star or the square or the triangle or any other design, and we will cover a leaf, cover a portion of the leaf using that black paper. Now what is happening? This part above over here, the part inside and the part over here, these parts are exposed to light, so they are receiving the sunlight, but the part of the leaf that is covered with the black paper, they are not exposed to the leaf, right? Sorry, they are not exposed to the light, correct? So now in that case, after 4 to 5 hours, we'll pluck this leaf, we will perform the iodine test over it. So when we'll perform the iodine test, we will find it out that we can see the black cover only in those areas where which were receiving enough sunlight, but the area which was not receiving sunlight will show the brown coloration. So this shows that sunlight is essential for photosynthesis. Fine, easy to understand. Now let's see our next experiment. Now we will talk about the experiment number 3, where we need to show that carbon dioxide is necessary for photosynthesis. So we spoke about chlorophyll, we spoke about carbon dioxide, sunlight, and now we are talking about carbon dioxide is essential for photosynthesis. So again, we'll perform some experiment where we will we'll take a leaf. So half of the leaf will be inside this conical flask and half of it will be outside the conical flask, and inside the conical flask it will be like tight, and it has this potassium hydroxide, and we know that what does this potassium hydroxide do? It absorbs all the carbon dioxide that is present over there in that air, right? So all the carbon dioxide that was present inside this conical flask was absorbed by the potassium hydroxide, but the leaf which was exposed to the air was getting enough carbon dioxide to perform photosynthesis, right? So we can also do it this way. We'll take a beaker, put some potassium hydroxide over here, half of the leaf is inside the flask and half of it is outside the flask, right? So then there will be we'll keep it in sun for some time. So before that, what do we need to do? We need to take a de-starched plant. What type of a plant we'll take? We'll take a de-starched plant. Then we'll start performing this experiment. And now after say 4 to 5 hours, we will take this leaf, and when we will perform the iodine test, we will find out that only the portion that was outside the flask or outside this beaker turned blue black, and rest the part that was inside the flask or the beaker, it did not turn blue black, it was brown in color. Why? Because this portion of the leaf has enough carbon dioxide, it has sunlight, it was getting water. So in that case, this portion of the leaf performed photosynthesis. But coming on to the inner portion of the leaf, so this portion of leaf was getting sunlight, it was getting water, it had chlorophyll pigment in it, but there was no carbon dioxide. So this shows that carbon dioxide is essential for photosynthesis. Now the experiment number 4, which asks us to show that oxygen is produced during photosynthesis. So how do we know that oxygen is produced during photosynthesis? So in that case, we'll take a water plant, say we will take hydrilla, right? We'll take a beaker full of water, we'll place it inside that plant in a pond water. So this beaker is full of pond water, we'll place this plant hydrilla in the pond water, and over it we will support it to keep the funnel off the bottom, right? So basically we will keep a funnel over this plant, and now what we will do, we'll take a test tube which is full of water and invert it over this funnel. And remember, the level of the water in the beaker should be above the mouth of this funnel. Okay, now after some time we'll keep it in a in sun for some time, and after that we will observe that there are some bubbles that can be seen inside this tube, right? Inside this test tube. So this, when we will test this gas using a splinter, it will burst into flame, as this is an oxygen gas and oxygen is a combustible gas, right? So it catches fire, correct? So this shows the presence of oxygen as a byproduct of photosynthesis. So when photosynthesis is performed, so that oxygen is released as a byproduct, because over here it has chlorophyll, it is receiving enough sunlight, right? And it has carbon dioxide also. So all of it has everything that a plant needs, a aquatic plant needs, and hence it is producing enough oxygen gas. Fine. The next topic is the importance of photosynthesis. So we are talking about photosynthesis, and we clearly know what is the importance of photosynthesis. First is food and second is oxygen, right? So talking about food, so talking about the food chains, talking about different animals, if we start, so all the organisms, directly or indirectly, they are dependent on plants, right? So these are the producers. So let us talk about the different herbivores. So there is a grasshopper, there is a rabbit, there is a mouse, right? All of them, they are dependent on plants, so they eat plants or their products, and then they are further eaten up by some carnivores, so secondary carnivores or the tertiary carnivores. So talking about any food chain, so take it as started with grass, then rabbit, and rabbit will be eaten up by the tiger. Talk about a carrot, carrot will be eaten by a rabbit, then that rabbit will be eaten by some another, say lion, right? So all the there might be different organisms in the food chain at different levels, but the primary level, the producer level, there is no change. Every time in the producer level there are plants. So plants are the producers, and without them there is no food. Whether the animal is a plant eater or that animal is a, say, another animal eater, right? But the major source of food is plants, right? And how do they give us this food? They perform photosynthesis. Now talking about the next topic, that is oxygen, right? So obviously they provide this free oxygen which is present in our atmosphere, and this free oxygen basically helps us to breathe in, perform all the cellular functions that are happening inside our body. And it is believed that scientists experimented, and they believe that around some billion years ago there was no life on the earth, because there was no oxygen on the earth, and this life started with the presence of plants. And when these plants started photosynthesizing, so that then it started then they started releasing oxygen, and hence they started the coming of animals onto the earth. The survival of animals became possible on the earth. Now talking about our next topic, that is carbon cycle. So what is this carbon cycle? So basically there are series of steps, series of chemical reactions that are occurring in the atmosphere, where carbon as an element is taken up by the living organisms, it is being processed, and finally it is returned back into the air. So that is the carbon cycle. So this carbon cycle has various steps. So it starts with photosynthesis, then comes respiration, then food. There are different food chains. There is respiration, there is decay of dead and decaying organic matter, there is combustion, and there is heating of limestone. So these are the different steps that happens during the carbon cycle, and let us now discuss each of them in detail. So the first process is the process of photosynthesis, where the green plants or the producers, they take carbon dioxide from the air, they perform photosynthesis, and they make they convert this carbon dioxide in the form of carbohydrates or sugars or starch, right? Now in the second step, this starch, sugar, or the food is being given to different kinds of organisms. So animals, they consume this food, right? Now through food chains, this carbon moves to different organs, correct? Then happens the respiration. Now this food goes into our body, where this glucose is being oxidized, and in turn carbon dioxide is being released into the air, right? So this happens during respiration. Then comes the decay. During decay process also, different microorganisms, they act on this wood, these dead and decaying matter, and where they release carbon dioxide into the air. So this carbon is again given back to the air. Then comes the combustion. So during the combustion of wood or fuels, carbon dioxide, carbon is released into the atmosphere. Fine. Now so these are the different sources where the carbon dioxide is being released into the atmosphere. Now heating of limestone. So during this fossilization, where fossil fuels, coal gas, oil, or this gas limestone, all of them are used. So while we heat them in those big furnaces, so in that process also carbon dioxide is being released into the atmosphere. So basically there is only a single way where we are actually trapping this carbon, and we are releasing it into the atmosphere in different ways, right? So they are trapped using the process of photosynthesis. When plants, they perform the process of photosynthesis, and rest in all the cases, this carbon, talking about the food chain, where it is this carbon is distributed to different organisms. Now through respiration, decay, combustion, burning of the limestone, so in all these processes this carbon is being given back to the atmosphere, right? So this is the carbon cycle, which is also the series of chemical reactions which are taking place, in which carbon is being taken up from the atmosphere. It is being used by the living organisms, it is being processed, and finally it is removed back into the atmosphere. So this is the carbon cycle, and these are the 6 main steps of the carbon cycle. So now, since our chapter is complete, there is a time to talk about some of the questions. So the question number 1 is given below is a schematic diagram to illustrate some aspects of photosynthesis. So fill up the gaps in the blank spaces by writing the names of the correct items, right? So this is what is being shown, that is the photosynthesis, right? So what is this? What is coming in this form? So this represents what? Sunlight. So this is sunlight. Now what is being given out from here? So what is given out? That is the oxygen gas is given out during photosynthesis. So using this chlorophyll, sunlight, water, so what is being formed? And also the carbon dioxide gas, glucose is being formed, right? And now what is this fourth one? So what is it? So what is happening? There is the movement of water through xylem tissue. Correct. Now the next one is, what are the phenomena do the thick arrows A and B show? So this is the arrow B, this is the arrow A, right? So what does this show? So this is a leaf, something is going out from the leaf. So what is going out from the leaf? Water vapor. So what is this process called? So this is called as transpiration, right? And when there is the movement of water, right, the movement of not water, the movement of substances upside and to the downward side. So what goes up and down, that is the bidirectional movement. So that is the food. And what is that process, which is the movement of food from leaves to different parts of the plant? So that process is called as translocation, right? So what is it called as? It is called as translocation. Now the next comes as the question number 2. The diagram below shows 2 test tubes A and B. Test tube A contains a green plant, test tube B contains both green water plant and a snail. Both the test tubes are kept in sunlight, right? So this is our experimental setup, where this is test tube A, this is B. There is a green water plant, there is a green water plant again, and along with that there is a snail, right? So the first question is, name the physiological process that releases the bubbles of oxygen. So what is that process? So that process is photosynthesis, right? And what is photosynthesis? So photosynthesis is a process by which green plants make their own food using carbon dioxide from air, water from the soil, and the chlorophyll, which is a green pigment present in plants. It traps solar energy and hence converts carbon dioxide and water into sugar and releases oxygen gas as a byproduct. Correct? So now question number 2, second part. Explain the physiological process that is mentioned in 1. So I have already explained you that physiological process, that is photosynthesis. So next is, what is the purpose of keeping snail in test tube B? So why we have kept the snail in test tube B? So the reason for keeping this snail is this snail will respire. So when this snail will respire, it will it will give out carbon dioxide, and that carbon dioxide will be used by the plant to perform photosynthesis. So this increased concentration of carbon dioxide in test tube B will increase the rate of photosynthesis, and hence more bubbles, more oxygen gas will be released in this particular test tube. Now the fourth question is, why does test tube B have more bubbles of oxygen? Why this has more bubbles? Because there is a snail is respiring, so that snail is giving out carbon dioxide. So carbon dioxide is being used by the green plant, so the rate of photosynthesis has increased, so there is increase in the level of oxygen concentration also, so more oxygen is being given out by the plant. So in that case, there are more number of oxygen bubbles that are being seen in the test tube B. Fifth, give an example of a water plant that can be named in the above experiment. So which water plant? Which water plant? Very simple one. We have already studied. That is Hydrilla. Right? Which water plant? Hydrilla. So let us start with our question number 3. So our question number 3 says, the diagram given alongside represents an experiment to prove that the importance of factor in the photosynthesis. So answer the questions that follow. So the question number 1 is, name the factors studied in the experiment. So this is the experiment where there is a potted plant, and this a portion of a leaf is covered with a black paper, right? So what factor of photosynthesis we are studying? So we are talking about sunlight. We are experimenting the presence of the importance of sunlight for photosynthesis. So next is, what will you observe in the experimental leaf after the starch test? So when we will perform the starch test on this leaf, the exposed area, so this area which is exposed to light will turn blue black, but the area which is covered with this green sheet will not turn blue black, it will turn brown, right? So the next is, explain the process of photosynthesis. So how are we going to explain this process of photosynthesis? So this process of photosynthesis is very important, right? And we are talking about it since we have started the chapter. So carbon dioxide is being trapped by the leaves, taken in by the stomata from the air, and water is absorbed by the roots. So in the presence of sunlight, right, and chlorophyll, which is a pigment that is present inside the leaf, it makes glucose and release some amount of water also, and oxygen is also released as a byproduct. So this is photosynthesis. Now we need to balance this equation that you will do. I know you know how to balance the equation, right? And this is the fourth question that is being asked. Give a balanced chemical equation to represent the process of photosynthesis. So now the question number 4, give a suitable biological reason for this. All life on earth is supported by photosynthesis. So we have to give a suitable biological reason. So that is true. If we talk about photosynthesis, it is a very significant process which on the earth which gives food directly or indirectly to all the living organisms that are present on the earth. So if there is no photosynthesis, there will be no food and there will be no oxygen. So this food and oxygen is very much important for all the living beings to survive. So hence we know how important is this photosynthesis to us. Now, next question, question number 5. Give suitable biological reason for this. All the food chains begin with the green plants. So, all the food chains, they do begin with the green plants. So, if you talk about the green plants, so green plants are basically what? They are the producers, right? So, these producers, they are taken up by, say, a rabbit, right? And this rabbit is eaten by, say, tiger, right? Now these producers, they are also taken by grasshopper, and now who will eat this grasshopper? Say a bird is going to eat this grasshopper, and now who will eat this small bird, right? So there will be a large bird who will be eating this, correct? So if you talk more and more about these, so say the producers, these producers will be eaten up by the deer, and this deer will be eaten up by the tiger. So basically all the food chains, they begin with the producers. If there are no plants, none of them will get food, right? So it is not like that only herbivores will suffer, but totally all the living organisms on the earth will suffer if there are no plants and if there is no food production by the plants. So now the question number 6. Question number 6 says a potted plant with the variegated leaves was taken in order to prove a factor necessary for photosynthesis. So the potted plant was kept in the dark for 24 hours and then placed in bright sunlight for a few hours. Observe the diagrams and answer the questions, right? So we took a potted plant with a variegated leaves like a croton plant, right? So it was kept in dark for 24 hours, then it was kept in sunlight for some hours. So what aspect of photosynthesis is being tested in the diagram? So over here we are testing chlorophyll, right? We are not covering it with anything. It is just kept in dark, then kept in sunlight. So what we are doing? We are talking about the essentiality of chlorophyll for plants to perform the process of photosynthesis. Now, the second part of the question, represent the process of photosynthesis in the form of a balanced equation. So students, these are the very, very important questions that are being given in your books, that are being asked in a previous year question. So this question has been asked second time, where you need to write the equation of photosynthesis. So it is going to be carbon dioxide plus water in the presence of sunlight and chlorophyll. It will give a glucose, some amount of water, and oxygen gas will be evolved. And now this equation has to be a balanced one, and I know you know how to balance it. You have studied that in chemistry, right? Now the third part of the question is, why was the plant kept in the dark before the beginning of the experiment? Why we have kept it in the dark? So as to destarch the leaf. So what does it mean by destarching the leaf? Making the plant free from starch. There is no more starch present inside the leaf. Now keep it in the sun. Now there is there is carbon dioxide, there is water, there is sunlight, and there is chlorophyll. So now that starch formation will start, right? So we can easily test when we kept it in the dark and when we kept it in the sun. So what are the differences in the plants, right? So to ensure that leaves are free from starch, so that is the reason why we kept that plant in the dark. Now the next is question number 4. What will be the result of the test performed on leaf A shown in the diagram? Give an example of the plant with the variegated leaves. So what will be the result of the test that is performed in the leaf A? So the leaf A, when we will perform that starch test, we will see that initially it will be pale yellow, because when there is no sunlight, that chlorophyll will also degenerate, right? It will also lose its color, and when we will perform the iodine test, we will see the golden brown color on the leaf, not the blue black color. Okay, now example of variegated leaf is the croton. Croton is the best example of the variegated leaves. So now the question number 7, complete the following para by filling in the blanks. To test a leaf for starch, the leaf is boiled in water to kill the cells. Right, so we are killing the cells when we are boiling that leaf in water. It is then boiled in a methylated spirit. So when it is boiled in a methylated spirit, why are we boiling it in a methylated spirit? To remove chlorophyll, right? Now leaf is dipped in warm water to soften. Now we are putting that leaf in a warm water so that it becomes soft, and then it is placed in a petri dish and dash solution is added. Which solution? Iodine solution is added. Now the region of the leaf which contains starch, it will turn blue black, and the region that does not contain starch will turn brown. Right? Simple. It was just the iodine test that was given in the form of the blanks. So students, I hope this chapter is clear to you. All the questions related to this chapter are clear to you. So now we will meet in our next video where we will talk about the next chapter. And till then, as you know, Learner Hub free है पर best है.